Thank you so much to the organizers for this workshop. It has been truly fantastic. Speakers feel obligated to say this, but I really mean it. It's been really, really amazing. And it's been the breadth of what's here has been really enjoyable. So thank you very much. Today I'm going to tell you about I have two stories. I imagine I will only get through story one, but I want to highlight the people who did the work, and I'll show some more pictures. But story one was largely from really amazing conversations with Ale Monguera, who was a postdoc with Olivier and now has his own lab at UCL, and Alex Grigas, who's this amazing postdoc. I was lucky enough to recruit to Syracuse to work on this project, and Rajendra, who's also an excellent postdoc, mostly works on something else but contributed to the project. And then if I have time, I'll tell you about a story with Andrea Liu, spearheaded by Sajjad Arsash, another fantastic postdoc. I've been really lucky to have these fantastic people. You should hire them if you see them. Okay. So the story today is, as you already know, I'm preaching to the choir. Biological tissues are amazing. And it was really nice to see the talk this morning, which focused a lot on structure and nanostructure. And then we saw some talks previously in this program about large-scale tissue flows, like the shape of skulls or the shape of teeth. And the idea that I wanted to emphasize today is where I become very interested and think we might have something useful to say is in thinking about what are the emergent mechanical properties that enable these flows. So how does the structure at the scale of a tissue facilitate things like flows and shapes and morphogenesis? That's the big question in my lab. Tissues are made, if you think about them as a material, they're simply an amazing material. They change their global shape to enable tissue function, including things like expansion and shear, like body elongation, but also things like localized sources and sinks at exactly the right place at exactly the right time. They open up holes. They have predetermined curvatures. We cannot make materials that do those things, obviously, yet. And while they're doing all of this amazing reconfiguration, in many cases, they maintain a specific small-scale cellular structure, which is important for cellular signaling, and they do things like maintain tissue integrity, like tension or stiffness or things like this. Also, at the same time they're doing all of those things, they're making cell fate decisions in certain pockets in order to generate specific different things that they're going to do. And they also adapt to perturbations that are out of context perturbations, like you shine a laser on the thing, which never happens in evolution, and somehow it manages to recover very well from such a perturbation. Okay. And one of the things that I'm interested in thinking about, especially more recently, is, how do they do all of this without an external processor? Many organisms have brains or nervous systems, but a lot of the time these decisions at the tissue level are made somehow via mechanical programming inside the tissue or biochemical programming inside the tissue. So how does that work? All right. So story one, which I'm assuming is most of what I get through today, and please, that means that I would love for you to ask lots and lots of questions during, is how can I have a tissue flow and maintain rigidity at the same time? As a material scientist, you don't usually think about those two things as being compatible with one another. And can a tissue program desired patterns of deformation, and why and how does that work? Okay. So Ale came to me and showed me this movie because I have worked a lot on epithelial layers and tissues that are at packing fraction unity, and we've done a lot of work on that. And he was like, look at this crazy tissue. So this is chick presomitic mesoderm. Even from this simple movie, what you can see, I'll try to play it again, is that there's dynamically remodeled contacts via made and broken cell-cell contacts here. You can see those. The cells don't have simple shapes. They seem to change their shape a lot in this tissue. And the tissue, but looking at it, zooming out a little bit, the tissue seems to me remarkably robust. There's not huge density fluctuations in this tissue. And the behavior seems kind of uniform in this patch. There's gradients along longer patches, but in this patch, it seems kind of robust. Oh, that's a good question. Olivier, do you know how long this movie is? I think it's a couple hours. Well, because I have another movie that where the cells don't remodel, and I know that one is 30 minutes. And so to get this level of remodeling, sort of, I think it has to be a little longer than tens of minutes, but maybe I'm wrong. I will look and tell you. I'm sorry, I should know that. Yeah, tens, and maybe an hour.
It's not, yeah, something like
Yeah,
that.
yeah. Okay, I should know the answer to that. Sorry. And and also I'll say, like, as a theorist, it sort of looks like these like cells are kind of pulling on each other, and like maybe it seems like they're under tension. Yeah. Oh yeah. So I will show you a bigger. Can you hold that question for? And I'll talk about the structure of this overall tissue in just a second. So just hold the question if I don't answer it. Is there another question? Okay. Okay, and so from a theoretical perspective, like this seems like, to quote another friend of mine, a hot mess. Like, how would one try to model this in some way that's predictive or useful is the question. And when Ali first showed this to me, I was like, I have no idea how to do that. And the reason why, in part, is because it looks quite different. So there's zebrafish presomitic mesoderm, and this is a picture of that from, this movie was from Ali when he was working with Oche Campos, and this is a beautiful model out of Oche Campos's group, Sang-Woo Kim, that is a model for this type of tissue. The cells are densely packed. This type of modeling is straightforward. Our group does something like it too. But it's really the structure I would claim, even though these are both functionally doing similar things, they look very different at the structure of individual cells, and even as the tissue. Yeah? Okay. Okay, so here's the answer to your question, which is this is the chick presomitic mesoderm, and so there's sort of notochord, and there's this region where this movie was taken from. Oh, I got to play the whole movie for it actually to be in the right spot. Sorry, I messed up. Okay, here is the movie. All right. And you can see the notochord structure, and then the presomitic mesoderm is sort of here that's going to eventually form the somites. Does that answer your question? And so there's a structure here that you can see. That's what you're looking at, is the higher density region. Then there's some interesting boundary condition out here of a different tissue type. So the boundary condition could be very important, and there are changes in density. So we're focused on just this little patch here. Yes?
To imagine the cells crawling on some extracellular substrate?
That is a great question. So very clearly, there is a soft extracellular matrix in the system composed largely of hyaluronic acid. But it seems that it's not like a rigid, like fibrillar ECM like you might see in other tissue types. In fact, what this reminds me of very much, again, just like taking a zoomed out picture, is like cartilage, if the cartilage part, like the sort of fibrillar part of cartilage, was instead made up of cells. So there's a cellular network embedded in this very soft ECM network. And, you can ask, that ECM network seems to be remodeled. So there was a talk by Kangxi where you sort of highlighted that those sort of soft networks are sometimes viscous and sometimes elastic. They can be viscoelastic. It seems that if you track these things, those ECM is remodeling enough that you might not think the cells could really pull on it well. And so it looks like they're crawling on one another, and I'll come back to that in the modeling. But it's a little bit of an open question, really. Okay, and so of course what you can do is you can do some nuclei labeling and look at, like, what is the dynamics of this tissue? What is the macro? So in my lab, we often try to connect like microscopic structure and dynamics of individual cells to macroscopic emergent dynamics of tissue. So what is the macroscopic emergent dynamics of this tissue? And so you can look at nuclear labeling, and then Rajendra did a fantastic job of doing some tracking in this system in 3 dimensions. And so I'm just showing you a 2-dimensional projection. And you can see that there's definitely some macroscopic flows in the system which have been well characterized.
What's the label? I mean, this is a transgenic chicken from UK?
Yes, that's my understanding. I am new to this modeling and to this system, so that's why a lot of my answers are not, so yes, but I don't know exactly. All right. It's already been shown by other folks that there is sort of this random motion. So this is the mean squared distance between neighbors. To get rid of the global flows, you could just look at how far apart neighbors move. And you can see that on long time scales, it's diffusive or fluid-like. If you look at the log of the squared displacement between neighbors as a function of the log of time. Or you can try to make networks of cells that are nearest neighbors with one another and ask what fraction of neighbors disappear as a function of time. But this has already been seen in other work. And actually, Maha and collaborators have this nice model that suggests that you could generate this type of behavior with just simple Brownian motion of the cells, that the cells are fluctuating randomly. And maybe that even, I told you I'm not going to be interested today in the gradients, AP gradients in the system, but you can even explain some cool AP gradients in terms of a gradation of cell motility in this type of model. Okay. But as a person that thinks a lot about cellular structure and materials, it looked like they were under tension. And so you can do a laser ablation experiment in this system. So you can put like a cut in this tissue in the embryo. And what you see, and this was a very quick PIV, so I'll show it to you again. But basically what you see is there's really a big burst of velocity, retraction velocity after you sort of slit it, which suggests that the system is under tension, at least on short time scales. And so you can, well, let me show you the movie one more time since that went by fast. Okay, here's the movie. So there's a big initial burst and you can quantify the retraction velocity. Yes, Suraj.
It seemed to be at an angle. Pattern is that, or is that just my eye?
So I think that we looked at sort of the orientation of this relative, and it seems like if you average over the whole thing, it's roughly, you know, it's approximately this. We have replicates of this, and they don't all look slanted in the same way. So there's a little bit of a variation, possibly in the tension, but not enough for us to quantify, at least the way we did here. Okay. And so you can quantify the retraction velocity. So it suggests that this network, the cellular network at least, does seem to be under tension. Okay. And the other thing is that you can look, I said, well, to my eye as a materials person, it looks remarkably uniform. And so one thing you can do is take this structure and ask, like, how uniform is it? And should I be surprised by that? So the way we chose to do that is we chose to normalize by the sort of texture of a random sequential addition pattern, if you know what that is. It's just a way of saying, okay, how would I expect random non-overlapping spheres? How would I expect them to be organized in space? And is a pattern more or less uniform than that? It's a measure of how uniform the packing of the cells are. And so what you find is the nuclei are more uniform than you would expect from a random packing, or RSA anyway, packing of spheres at the same density. And I'll come back to why that's important later. Okay, so we have a preprint out on this now, but we're in the process of thinking about it, so I'd welcome any feedback. So there's remodeling of these contacts very regularly, and yet the network is under tension. You have a question? Yeah.
It seems that the cells are not in contact with each other. It seems that they're freely moving. It's not like epithelial cells where they have this solid behavior, right?
Yeah.
And then when you do the laser ablation, because the cells are not, the neighboring cells are not touching from the video, I think.
We think they're, we have some pictures in this preprint where we've done mosaic labeling of the cells, and it really looks like there's these sort of extensive protrusions of each one of these cells. So even though their cell bodies are largely not touching, it looks like they're making protrusions and contacting at cell-cell contacts at like, almost like a star, a stellate system.
When you do laser ablation, breaking those contacts, or is there any?
Yes, because we're totally ablating a pretty good chunk of it, so we must be destroying those contacts. We make the ablated region large enough that we're really getting rid of those cell-cell contacts.
Do you think you also update?
We could, yes, we must be, right? And so the question is how solid-like is that hyaluronic acid underlying ECM? Again, if you look at the dynamics in one of these movies, because the cells are remodeling on timescales of 10 minutes and really changing the shape of those pockets with no cells, it seems like that can't be acting as like a rigid object on timescales of this remodeling. But open question.
I'm just thinking about examples in cells that can move through pretty rigid stuff in a very similar way, right? And the way they do that is that they just go through the pillars or through the pores, and so effectively looks like the matrix is not there, but they can deform. So I'm wondering if you have a way to distinguish if it matters at all. I don't.
Yeah, so one thing you can do a back of the envelope calculation, which is like at this concentrations of HA, like what's the pore size in the HA network, and it's super small. So like they have to be either like degrading, you know what I mean? They have to either have to be like moving it around or doing something. And I agree that's a completely open question. And what I'll tell you is what we assume for the rest of the thing is that it's possibly facilitating a lot of the interactions. It could be even facilitating the cell motion and motility. And we know that it is either acting as a signaling molecule or as a mechanical molecule that's like HA is definitely a facilitator of motility. But we're going to treat it right now as if that HA is not like, cells, the way that we'll model it, cells could be crawling on it. I have nothing to say about whether it's required for their crawling or not. The question that the assumption that we do make is we haven't included its mechanical, like sort of pressure on this cellular network. That's an obvious next step. Yes.
I think this in a more like a percolation theory point of view, basically saying that is there a
Percolating rigid tension network? Yes, hold that thought if you don't mind. That's exactly what I will talk to you about. Yes.
So I guess in a theory agnostic way, if one were to do the laser ablation kind of in different regions of an embryo or over time, would one see a lot of different relaxation velocities, or there regions where one wouldn't see much one at all? Yeah, so we did. So actually the data in this preprint, which is like, we want to do a lot more. This is the first pass. We actually did ablations at different places along the AP axis, which ostensibly has different sort of properties. And the retraction velocity was remarkably similar. So we could average over it in different places. So if the viscosity, there's a problem, retraction velocity is a tension divided by a viscosity. So the viscosity of the medium, anyway, there's lots of questions about why it is so similar. And one can't automatically assume it's just because the tension is the same everywhere. But I will tell you that it seems very similar across different regions of the chick presomitic mesoderm. Okay. Yeah, this is the first time I've ever talked about this. So you're getting the raw version, and the questions are very helpful. Okay, so actually at the same time, like very independently, I collaborate with Andrea Liu. I did not know she was doing this. That happens. And so at the same time we were doing this, basically John Crocker and Liu and these fantastic trainees had this idea. They asked the question, what properties of an actin network would be required to allow you to have a dynamically remodeling network that was changing its neighbors all the time, and yet still under tension and maintaining mechanical rigidity? And they called this rigidity homeostasis. So they have a different word. We call this fluid under tension. They call it rigidity homeostasis. But I think the question, and this is the big question for the rest of the talk that I would like, this is the mystery to me when we solve these things, is how can you make a network that maintains tension and still remodels? And let me tell you why, as a material scientist, like by training, it's not obvious. So it is obvious if you have, in my opinion, if you have a dense epithelial packing, because you can have these contractile units that we heard about in the system, and those can be generating tensions. But because it's filling all of space, those tensions don't lead to any, well, maybe some, but mild density fluctuations, not major density fluctuations because you're filling space. But, oh, yes.
The ablation, the laser ablation was done in the embryo or in the dissected?
In the embryo.
Because you could also imagine that the tension is coming from the superficial ectoderm and endoderm, I imagine. Right.
And so we do have plans to do it in explants. The challenge with the explants is we think some of the tension may also be generated from the boundary conditions in the in vivo system. So it's hard to dissect those two different not just the ectoderm above and below, but also
You know, in the system that Arthur developed, where he has these channels, these PDMS channels, and he lets the
Yes.
there you could probably do it.
We've had that exact conversation, in fact. So the short answer is yes, this experiment has that conflation that somehow that tension could also be generated by so one nice thing that we're thinking about doing to answer that precise question also is that it looks like when cells break contacts just naturally in this system, they retract a little bit. So we think that maybe we don't even have to do anything artificial to the system, that maybe if we could track individual cell retraction speeds that we could get at this tension as well. So there's evidence also that this tension that we're measuring, at least in part, is generated by the cellular network itself because of those videos, but we haven't quantified it, the difference. Yeah, okay. So the claim that I'm making is that you can have rearranging systems under tension if you're space filling. But if you're sparse, if your network is sparse, there's basically an instability that's hard to get away from. And the instability is that if you have bonds breaking, and you have a system, a network that's under tension, when you break a bond, then the system clusters, and then there's an instability that things tend to break and break more. And unless you have a way to make new bonds, these things that are dilute generally cluster. Okay, so here's a picture of it in like a spring network. If you have bonds that can rearrange, even if they're able to reattach, they start to cluster and then they're further away from neighbors and then they don't reattach. Okay, that's this. Okay, so what type of model? So we're interested in making a model of this tissue that can recapitulate the three key observables that we see in the experiment. One is the dynamics, which we characterize in terms of a diffusion coefficient, like how fast these cells change neighbors. Another is this tension, which is how much basically we want there to be a negative pressure. So we define it as a negative pressure in the system. Okay, we want the thing to be under tension. And we want the structure to be like the one we observe in the experiments, which is fairly uniform and not clustered. So how does it do that?
Sorry, that
Yeah.
ablation experiment, you're only looking at the recoil retraction of the cells.
The cells
You
themselves.
haven't marked the ECM itself.
No, and that's another thing that would be very interesting to do. There's this whole, I mean, now that we've seen this, there's this whole set of questions like what really is going on.
It's just more like what's the unit here? Is the unit is not the cell, the sparse cells alone, right? If they're all attached to the ECM and they have this turnover, then the entity you want, the viscoelastic entity, is the bulk of the cells with the ECM.
Right. And I would say one of the things that I'm inspired by is this cartilage literature, which doesn't have the remodeling aspect, but you can think about sort of the collagen networks as being load-bearing structures that sort of have the tension in them, and then the soft aggrecan hyaluronic acid structure generates like a hydrostatic pressure, but it's not really, it's sort of supporting in some way that tension in the structure, but there are these two independent pieces. And the reason that I think it might be appropriate, so we made that approximation here. The reason I think it might be appropriate is because you really do, when these cells are changing neighbors and remodeling on this time scale of tens of minutes, they really are the extracellular matrix in there is soft, and it seems to be getting moved around by the cell motion quite a bit. So it's kind of the comment would be is at least on timescales that commensurate with tens of minutes, that thing has to have an effective viscosity, I would argue, because it's being remodeled at the same rate that the cell contacts are being made and broken, because it's not the cells moving through the pores because the pores are way too small. Yeah. Yes. And that would be very interesting. We haven't done. Okay. Yeah. Yes.
You know, we did a while ago labeling the fibronectin with an antibody coupled to a red fluorophore.
Yes.
And making movies, and it doesn't really move much around. So when you measure the diffusion of the microfibrils or the globules of fibronectin, you don't see that most of the movement is advection, basically.
Remember that?
Yeah,
I wanted to do that.
Yeah, and I think there's also a question of how much is because the fibronectin networks, as far as I understand, are not very dense in this system, right? They sort of seem ephemeral, to use like a non-scientific word. And so it's unclear to me how much structure that fibronectin is imparting in a system like this, or if it's just sort of,
Posteriorly, where there's a lot of movement, probably not that much.
yeah, where the diffusion is high, like we're reporting here, because we're not in the, anyway, I would love to discuss in more detail. The assumption we've made here is that fibronectin is not playing a role other than possibly being a scaffold for cell crawling.
But you can, it nevertheless says that the matrix is probably not completely moved around.
Oh, I don't mean large scale flows of the matrix. I just mean when the cells are remodeling their contacts, that the local soft structure is not sufficient to prevent them from doing that remodeling. Sorry, that was unclear. I'm not saying there's large scale flows, but I am suggesting that there must be small scale local flows of that soft ECM, and that on sort of this time scale where the remodeling is happening, it's not acting like a solid that's preventing a change in the state. Yeah, okay, great. Okay, cool, so we wanna do this. Okay, and so the idea that Alex had, so originally I was like, oh, we should make these like star polymer-like model with arms and like something complicated. And Alex was like, no, we should not do that. Instead, he's super clever. He came up with this super cool idea, which is that, well, if I just only make arms between particles when cells touch, but the arms are hysteretic in the sense that once I make a contact, I keep it for a lot longer when those particles separate, I get arms for free, like arms that are touching for free. And it's a much simpler model with a lot fewer parameters to investigate. We actually did do the star polymer thing, by the way, and it was horrible for reasons I won't get into here. But this is what we ended up with, and it works. I'll show you. It's remarkably simple, but works remarkably well. So just to give, for the physicists in the audience, to give you an idea of what the potential is, it's basically like particles don't interact until they touch, then there's a repulsive spring, but the weird thing is that now there's a hysteretic, so if they pull apart, they keep the spring, and then the key cellular parameter that we think is relevant for the biology is there's a characteristic rate, which will be important later, K off, at which you can break these hysteretic bonds, and we view that as a cellular parameter, that basically cells like are, we don't know whether they're contacting via cell-cell contacts only, or if it's cell-ECM cell contacts. We're agnostic about that too with this scale of model, and we'll just see where we get, but this will be a key parameter.
Sorry, the spring is always repulsive.
No, here it's attractive. So it's under tension here. It's like trying to get at these tensioned arms. And so we're basically allowing these tensioned arms to form when cells make contact, but then they keep them even when they're under tension, but they lose them at this K-off rate. That's the simple model. Yes?
Does this K-off rate depend on the tension?
Great question. We've investigated lots of options. For what I'll show you here, we've made it not a catch bond or a slip bond. It's a straight up rate that's independent of the tension, and that's a modeling choice. And I think it's important. Okay, so it's very unusual. Okay, so just to say, like, if you had like a Leonard-Jones interaction or some standard physics interaction, the more you pull on it, you'll sort of make it easier for the bond to break. And so this off rate would naturally depend on the tension in the bond. There's a paper that sort of does this standard. There's catch bonds in which that you get stronger as you get under force. There's slip bonds where you get even weaker than Bell's. And so what we did here is something that's a little bit closer to a catch bond, and that we said, okay, we're going to make this independent of this off rate, independent of the tension instead of dependent. Yeah.
The signal for the spring is not the same, not equal to basically two times the radius. It's actually the
Oh, you're asking the rest length for the spring. That's something that we can actually vary in the model. Here, what we've made it is we've made the rest length for the spring where they touch. And so by choosing the radius of the particles, we've chosen a characteristic length scale in the problem, which is, you may say, it's the characteristic scale over which cells touch as an initial assumption.
And they can make only one spring at a time?
No, I'll show you, you can make as many as you want. Okay, yeah. Okay, so this is what the model looks like with lots of spring interactions. But if we just do this hysteretic particle model with remodeling, you'll be unsurprised to learn that you just break a bunch of bonds and the system relaxes. Because if you allow the system to break bonds, you know, even if you don't have a rate that's dependent on tension, you're still going to put things under tension and then things will cluster. So this clusters. Yeah.
If this case, it's just overlap dynamics, there's no motility, or
I will tell you, this is none, and I'll tell you in a second exactly what you're going to ask.
and you don't put any tension.
Periodic boundary conditions, no, and so there's some tension initially by the density of these objects, and there's some way of preparing them. Everything I'll tell you about with activity has a steady state that doesn't depend on the initial conditions. This does. But that's not what we're mostly, I just wanted to show you that it clusters if you don't put in any activity. So what Suraj is probably getting at is that you need to inject energy to establish contacts. And so the obvious way to inject energy to establish contacts to a physicist, and also based on a bunch of other previous modeling work, is to have a self-propelled particle, or basically like a cell that has a direction it wants to migrate and a persistence time. And that's because you could imagine, well, if I have a persistence time and I have, an adhesive interaction to my neighbor, I can generate tensions by walking this way in the spring tightens in the back, for example. Okay. The interesting thing to me is that this does not generate the network we want. So if you just have self-propelled particles, and we looked, I'll show you in a second, over a huge parameter regime because we thought this would work, it doesn't work at all. It always clusters, always. Okay. And I can tell you, and I'll tell you in a second why we think this is. Okay. But first I'll show you that we swept a very large parameter regime. So the parameters we're interested in is this off rate at which I, make and break contacts, and the magnitude of the active force I can generate by basically this self-propulsion. That's these two axes. So we can generate diffusion, no problem. That we can generate a system that remodels contacts. We can even make a system that generates tension. So in this bright yellow region over here, we can make a system that generates a lot of tension, but it always clumps. So what I'm showing you here is anything above 1 basically is clumpy, okay? And you generate, especially where you generate this tension, you're super clumpy. It comes along for the ride. Okay, so then, well, how else can we inject energy? And what is the biology really doing? What it tells you, I think, is that the biology is doing something smart. It's not just injecting energy in some random direction. And so what Alex realized is that, well, maybe there's a couple of different ways you could think about it. So you form these adhesions from contacts. So you make these adhesions, but maybe it's generating tension from crawling like a self-propelled particle. So it's generating tension from crawling, but maybe it's crawling away from neighbors. So those of you who are, sort of have seen it in the literature, not this particular cell type, but lots of cell types have what's called contact inhibition of locomotion, which is where you make a contact with a neighbor and you go in the opposite direction. You have a polarization machinery that crawls in the opposite direction. Okay, but as we've already discussed, it's not clear that this cell type is actually crawling on the ECM. And so we were like, well, how else could they be generating tensions? And another way, maybe they're making contacts with neighbors and exerting tension across cell-cell contacts, and they're kind of reaching out and grabbing someone and pulling. So we can look at that too. So adhesions from reaching out, and then you get some tension by ratcheting that spring. And the key ingredient that we put in both of these models is basically we wanted something like contact inhibition of locomotion, where we wanted you to reach out in a direction towards empty space. You somehow had to know about empty space and reach away from existing neighbors. Yes?
They're actively sensing something like and if each of the cell is a sink because they're absorbing it, then you want to move away from another neighboring cell because that cell is basically taking up all the stuff. There's a local minima, so you move away.
Yeah. You could think of lots of ways to create a rule like this. Yours is a chemotactic rule or something like this. In the literature on contact inhibition of locomotion, there's a lot there. But one of the things that you think about happening is when I make a contact with a neighbor, that there's some machinery that gets set up at that contact, and then there's row GTPases and sort of rack, and there's a whole biopathway that actually just automatically polarizes you in the opposite direction. So you could envision it something else just in cell autonomous that does it. Well, not exactly cell autonomous, but after a contact with a neighbor that goes in the other direction. Yes?
So when looking at the videos of the model, it seems like when the clustering that the cells get a lot closer together than when they're initialized, literally the distance between the centroids
Yeah.
changes quite a bit. You have all this negative space.
Mhm.
When looking at the videos from the ablations, it seems like that doesn't happen, but to me, and maybe couldn't happen because then like there's a volume that can't change to the cells, and I'm wondering, is that a relevant thing that needs to be thought about when trying to model this, or is it?
So is your question that there's ECM filling that space and it has to go somewhere, or that in the experiments you don't observe anything that looks like that clumping?
Yeah, in the experiments, you certainly you don't, but then to see all of the centroids of the cells in the video get like half as close, that seems like it wouldn't be possible, maybe. And so I'm wondering then, does that mean that this sort of model like couldn't possibly explain? Like,
yeah, the argument that I'm making, if you wish, the argument I'm making is exactly what you just said. This model can't possibly explain the experiments. You don't see those big patches. It would be the natural, maybe from a stupid physicist point of view, but it would be the minimal model, the null model that you would write down, and it doesn't work. And it doesn't work for an obvious reason, because there's an instability, even in the presence of injection of energy, there's a clustering instability if I'm under, it's very generic, and I'll explain why it's very generic, because it happens in actin too, as it turns out. But there's a clustering instability that if I'm under tension and I am sparse, then I'm going to have a clustering instability. So I need to do work against that clustering instability. And the argument is that contact inhibition of locomotion naturally does that.
Yeah. In cells, but in actin, you can, I'm not 100% sure, but you can prevent that if you have turnover.
Oh yeah, filament growing. And I will talk a little bit about Andrea's paper, and I'll point you to some details, because they included filament turnover, and I will talk, and it still doesn't rescue the problem.
So yeah, what would turnover look like for cells?
Oh, I don't know.
Okay.
It's a different, I'm not saying turnover happens in this system. I'm making I'm a physicist by training. I'm trying to make a sort of a generic statement, which is wrong in detail in a lot of ways. But I think it is a principle that might be useful for organizing, which is that if you want a tension sparse system, the point that I'm making is that's actually very difficult to generate microscopically, because there's a clustering instability associated with it very naturally, and we've tried, it's very hard to get away from that clustering instability. That's the big picture idea. And then there's all these details and like sort of important ones. I'm not trying to say they're not important, but like, oh, what if I have turnover? Like, what if I have this dynamics? What if, yeah.
I'm saying that you can rescue your model maybe if there was a feature like
Turnover.
Yeah,
it turns out that turnover isn't sufficient, well, at least in the actin system, turnover isn't sufficient to rescue it, but there's nothing like that here. Okay, so now I'm going to show you that if you add something like contact inhibition of locomotion, which we do in two ways. One is that you crawl away from neighbors using some yet known mechanism for crawling, or the other one is you just make new contacts away from neighbors and you pull on contacts away from neighbors. Either one we call contact inhibition of locomotion-like, but you can put them in the model, okay? And so I wouldn't be talking if it doesn't work. So it works. So, here's a movie of this type of model where as soon as you put in something like, I tend to crawl away from neighbors, then it really forms this beautiful network, and there's a robust region where it forms it. And so here's the phase diagram that shows it. Basically in this I think I have a build that shows it. Up here in this upper corner, you get a diffusive network that is also under significant tension, and it has a very nice uniform structure. Yeah.
Would assuming that the cells are incompressible have a similar effect?
Yeah.
With the clamps, you really reduce the distance between the cells?
Yes, exactly. So and that's what sort of my comment, epithelial cells don't have to deal with this problem because they're space filling. And so even if you pull on them, there's no huge density fluctuation that's allowed. So you can stay uniform and under tension kind of for free. Does that answer your question?
Yeah, but so if you formulated this instead of doing a contact inhibition type thing by saying that they have to maintain a certain cell radius.
Oh, yeah, it's effectively, the reason I answer the question, in my mind, if you basically make the cell, like an effective cell radius big, it's basically a big packing fraction that's it feels a lot like an epithelial model to my physicist mind. Yeah, yes.
Have you looked at how the parsing dynamics changes as you turn on this parameter.
Yes, it's super. There's all sorts of, I mean, Alex is probably going to write a PRE article about all of the physics of this stuff. There's a lot there. There's also a lot, like if you're a glass physicist, like there's caging and subdiffuse, there's all this stuff. There's a bunch of cool, I'd be happy to discuss in a lot more detail. There's a lot of cool physics stuff.
When they come in contact, the repulsion might not be even in contact inhibition, or locomotion. It won't be likely inflant and infront. There will be a delay before they start moving away from each other.
Yeah.
Have you quantified that time and have you incorporated?
The short answer is no. Because we're interested in doing it in the in vivo system, it's very difficult because like, imagine, okay, so the way we thought about trying to show this is by trying to say, oh, when I make a new contact, how long does that contact last? What's the motion? But because you really effectively have a lot of contacts in all directions in this uniform tissue, it's really hard, and the protrusions are small, right? It's really hard to be sure that you have a new contact. Is that contact effective in this way? One of the things that you can look at, I think, as a tell, although we're still in the process of doing this stuff, is that you expect there to be fluctuations where holes open up in the system naturally, in the natural system. And what you can see is that the holes like really close via cell-cell contacts coming into the thing. In order to quantify how different that would be from like an active Brownian assumption or something else, that's the tricky part. And how to really show that that would not be expected by chance in a system is how we've tried to do it. We're thinking about how to do that. But what I will say is what you do, the one thing that we do see is if a hole naturally opens up, you definitely regularly see cells coming in and making new contacts across that hole to fill it up.
So you described two ways to generate this movement away. So one is cells will migrate away from the cell they contact. The other is if they make a cell contact, they grab the other one. And you said those were equivalent, but intuitively it seems they're pretty different. Like if you had a hole with the two sides, it would expand. No. If you didn't have periodic boundary conditions, it would expand.
Yes.
Boundary conditions that would collapse if they're going.
So this is precisely, actually, you're thinking exactly, so this is precisely why I made Alex do the cell reaching model, is because if you try to do a droplet experiment on the stupid one with crawling, it's insane. Like it makes a droplet that like crawls out. Okay, and I was like, that's not realistic. If we want to try to model like droplet fusion experiments for this thing, doesn't seem to be swimming or crawling or anything in directions away from neighbors at the edges of a droplet. So this ratcheting model has very different boundary condition effects so I misspoke. They are similar in this one sense. So both of these models will generate, so this is, you can even see the dynamics are different. They look a little different even by eye. This is the reaching out and grabbing model. This movie looks I can tell which one is which by looking at it. The one way in which these two models are similar is they really do both have a region where you get this fluid-undertension with a uniform. They both work to fix the clustering instability.
Boundary conditions, one would work and one would clump.
Yeah, it does crazy stuff. But it doesn't clump. It actually has this, anyway, it has very crazy non-physical behavior. The other thing that I will say that you might be interested in is that what does happen in both of these models to some extent is that if you imagine coating like a microchannel with something that the cells can make adhesive things to, this model will do an amazingly good job of filling the channel and expanding into it and putting the whole thing under tension. So it's kind of like this, like from a material standpoint, it's a super cool material. Like we're talking to people who might try to we have some ideas of how to make this in a colloidal system. And like you could think of a system that would really like fill gaps and then contract the gaps. And it's hard to think of a remodeling system that could generate tensions uniformly in this nice way. And there's some applications for materials that could be cool. Okay, but okay. Yes, Suraj and then Adrian.
The point on colloidal gels, presumably, is that an analog of syneresis?
I don't know what that is.
When yoga, for instance, when you break the contact with the wall, collapses and permeates the filling fluid out and loses some tension, but don't lose rigidity.
Yes.
Is there a version of that?
I see. Yeah. So the short answer is I haven't studied that. I think this will do something even more weird. than a gel, because it will actually, like, both of these models will spontaneously fill empty space, right? The ratcheting model will fill it a little bit more slowly because it sort of has to pull itself along the boundaries, but it will. And it's better than a gel in the sense that it will spontaneously reform. It's like spontaneously self-healing of even catastrophic events like that. And there are many things that will self-heal. What is not clear to me is if that non-living or something with something like this will heal and then be able to generate uniform tension on a thing, which you could imagine would be super useful. I don't know, because I think this will happen to the clumping thing will happen to any other, even if you inject energy in it, it won't do this. Okay, I don't know. Yes, Adrian.
So I think the universality of this is so cool. So what is the actin doing that generates this inhibition?
I think I'll have time to get to that. Can you just, for a few minutes, I promise I will say that at the end. Yeah. Okay. So generates a uniform tissue. And it has like really interesting physicsy properties, but I won't go into that in this talk. There's subdiffusive behavior, there's all this cool stuff. But I guess the point is that you do have regimes where even these models cluster. So you kind of wonder, there's a lot of condensation things that are happening in these mesenchymal tissue types. So you wonder if maybe it utilizes a small change in parameters could give you a condensation or a clustering phase and not this uniform phase. So maybe if you take one thing away from this talk, I would say that like the uniform state is actively doing work against an instability. And so as soon as you turn off that contact inhibition of locomotion, you'll get condensation sort of for free in a model like this, just like you do in a gelating colloid, for example, which is kind of neat. You could think about it, what biology could use that for if it works that way. Okay, and here's the phase that matches experiments over here, this uniform state, and you have to sort of do work to get it. Okay. I don't know how much of interest this will be to this group, but just to say, it's kind of lovely and remarkable that you can, and this is all Alex. Alex's picture should be up here. You can come up with a very lovely continuum, very simple description, like a constitutive model of this system. And I'll just walk you through it very briefly. The key quantity Alex realized is the average contraction of a bond. So it's how much a bond contracts times how much time you have to wait for that contact to be unbound. Right? And the nice thing is that this, and you can do this integral and you get this equation, but from simple two-state kinetics, like if you have an on and off rate of bonds, you can actually write down what the fraction of bound in this type of system, what the fraction of cells that are bound to one another under a mean field assumption. And then the macroscopic tension is the product of this, contraction of a bond times the fraction bound, which you know. And if the contraction becomes motion later, then the flux of binding times the contraction will give you the diffusion constant. And so it gives you, and you know what the flux bound again by a two-state kinetic model. And so it gives you a way to collapse all of the data from all of these simulations with cell scale parameters that you could potentially even measure. Okay, and so here's what the data of diffusion versus this model. I'm showing you data for the ratcheting model, which is the model where the cells reach out and make contacts with neighbors and pool. And here's the diffusion constant versus the ratcheting rate. And this is the collapse that would be suggested by this continuum model. And this is, in fact, basically an analytic prediction for what this curve should be. And it matches pretty well with a big bunch of simulation data. Similarly, for the total global macroscopic tension in the system, this collapse was even better. Okay. This is the structural quantity, which we have no idea about. So you could do a phenomenological fit to this. We have some reasons for why it behaves like this, but there's a phenomenological fit here that I don't know how to predict from first principles, but you can fit it. And there's some reasons. We know what some of the things are. Okay, but what this means is we think, this is all brand new, so we haven't tested this yet, but we think you can measure these cell scale parameters, which is K off and K on, which is the rate at which cells make contacts, break contacts. The rate at which tension is injected at new contacts, we think there are some ways you could try to get at that by looking at sort of the growth of the length of arm contacts. And we do know you can measure like how much time it takes for the contraction. That's something you can look at a video and see. And then we can predict with those things, like without any fit parameters, a measured diffusion constant and a tension. It's kind of neat. And the structural thing has some additional fit parameters. But this is kind of cool. It's like a constitutive law for this weird type of tissue. It's a prediction of like the macroscopic behavior in terms of things that you could actually measure as a cell. And then you would start to think of, okay, like if I have sort of different regions of the presomitic mesoderm, or if I have different organisms that have these sparse mesenchyme, how are they changing these things which, you know, the cells have control over, and maybe I could even think of molecular mechanisms that are driving these things. How would I predict that it should change like the flows of the tissue or the tension in the tissue or things that are relevant? So it's kind of neat. Yeah.
Did you try to transpose this in vitro with these cells? Because contact inhibition, I think that's been first described in vitro a long time ago. So
yeah, so we're doing right now, we're looking at sort of explant spreading experiments because that's most easy to visualize these contact inhibition sort of type of interactions. And when you look at a video and you see these cell spreading experiments, they really do seem to come together and then go up. There's visual evidence and we can cherry pick examples, but what we're trying to do right now is in these explants get systematic statistical description of the contact inhibition of locomotion. Yes, so we're trying. Yeah, we're in process. Yes.
Sorry, how do you measure the on and off rates of the adhesion?
We would just take pictures, be like, okay, if we zoom in, because you can do sort of mosaic labeling. And so when a contact breaks, you see a snapping of the arms. And so we think we could get estimates of that. If you do full cytoplasmic labeling, you get some estimate of the rate at which these cells make contacts across protrusions as well. So we think we can do that just from image analysis.
Time to once they're actually close, and then seeing a further rearrangement of the membrane that makes it seem as though
yeah, I mean, I was thinking a much more zoomed out picture, like where you just like, or like, oh, I have these basic, like you would go in with like, you know, your favorite AI image analysis tool and you'd say, oh, these two cytoplasmically labeled bodies at like a low resolution are not touching, and then they are touching, and there's some rate at which that happens.
I see.
Something stupid, yeah.
What are timescales for those contact adhesion and the contraction?
We have not characterized them yet. This is new work. We have plans to measure all these things. I'm telling you, I think they can be measured, but we haven't done it yet.
Just like
a ballpark.
Magnitude.
Yeah. So here's this video again, which like I said, I think is on the order of about tens of minutes. And you basically see, like if you look, here's one, like there's a rate at which in that one between two frames, right, in this video, you could see it sort of come on and come off. So I think it's on the order of minutes.
And there's sorry.
Oh, sorry.
There's a critical density of cells with which this would work.
If they're able to crawl, or depending on how far, there's a parameter which says in the arm reaching model, how far can I reach before I can't reach anymore? It is like the cool thing about this is it's very robust to changes in density, actually. Very robust. Because as long as the cell can make a new contact somehow, either by crawling or whatever, you robustly get this rule, the set of rules leads to a tensioned network pretty robustly. And when you increase the tension in the network, they actually get a little further apart. You know, so the density interestingly, the density isn't the important thing, unlike most materials. Yes.
Following that, so is that currently the evidence for the tension comes from that laser ablation?
Yes.
So just thinking about a couple of, first of all, you could imagine a situation where, say, you use a fluorophore localized to the nucleus to be able to to kill the cells with light.
Yes.
In a way that doesn't damage the surrounding tissue at all. And then you could only remove cells from a band and ask whether you see the same, and would you predict that you would necessarily see the same contraction because the entire tension is mediated by the cells? And then maybe conversely, if you remove cells entirely, so that you sparsify them significantly so that they're no longer in contact with each other, you shouldn't be able to transmit tension across the entire structure anymore. And so if you carry out the ablation at that point, you shouldn't see a rebound.
Yeah, that latter one is something that we have thought about, because the question that would sort of help to answer the question about what's the role of this soft ECM network in generating mechanical properties. The first one, I think a challenge with it is that, you know, the zombie cells, like how long does their cytoskeletal, you know, there's some, if they've already made a contact, there's some tension being exerted across the cell-cell contact. I don't know what the time scale is for the degradation of that tension.
If you make it large enough, presumably you just have to break tension across an entire band over a period of tens of minutes.
Yeah, I might, so maybe we can talk later. I don't know if that would be as sharp of a result as something like an ablation. I might worry that there's a time scale there that would take a long time to resolve. Yeah, that, yes, that would be, I agree.
So if it turns out, just as a play, if it turns out that you do that, and you basically find that all of this effect is not explained by tension to the cells.
Yes.
Where does that leave this?
Yeah, so I think the question is that like I said, one of the things you do see is that when cells break contacts, you have like a very rapid, hard to believe was like a you know, like some polymerization situation of the cells retracting. So I think that even if there was some macroscopic, like, because actually like this hyaluronic acid could be generating a pressure that's generating a lot of the tension in this network. And if you like use hyaluronase or something, like you might, that's another thing we're thinking about trying, is to get rid of that, degrade that ECM network, and then look at, see what happens to the properties, which might be an easier experiment than some of these other ones. But I think that, I mean, if you really have zero tension at all across this network, then this does leave this in sort of a situation of like, well, what's the point? You can have a network not under tension that's robustly uniform, no problem. But I do think the observation, both that you have this sort of laser ablation that causes really a rapid retraction of the cell bodies themselves, and the fact that you see that when the cells break contact, that they retract, there seems to be some tension in the cell bodies. It's hard to imagine that phenomenology without something there. It might not be all of the tension that's supported in the network, I think. Yeah.
Is it at all feasible to embed in the HA?
Yeah, that's a good question.
Measure the deformation field that these cells are exerting in some way, right?
Yeah.
And then, you know, you would imagine that it's tension with like swimmers, right? Force quadrupoles, right? And that could be a signature that you should see in how your beats are flowing.
Yeah, I think that even what you said was interesting, even just trying to get a handle on what are the microscopic flows of this soft ECM as the cells are remodeling would in and of itself be interesting, even without doing any additional perturbation. So we haven't done it. I don't know how difficult it is to do.
Needs will diffuse inside of HHL, so you need to like attach it, but maybe like the HA binding protein and do speckle and things.
Depends on the size.
Yeah, yeah. It might be interesting just to try it as a, you know, see what happens experiment. Yeah, and then you, because the other thing that you might say is you might also be able to estimate what you would expect the diffusion to be, and if the motion is significantly larger than that during remodeling, that would also be an interesting signal. I think it's worth thinking about. It's a really good suggestion. Yeah. Yes.
At any time, I know, if you just look at the connectivity between the average number of degree, and if you see is there any sort of clustering, like for in the transitivity, if A and B are connected to C, then I'm sorry, if A is connected to B, B is connected to C, is A and C also connected? So if you form this sort of this thing, and at any point in time, the average degree in the system remains the same.
So the short answer is our data isn't good enough to reconstruct the connectivity network that you want, because this is three-dimensional in vivo data sets where the cell body, like there's these thin protrusions that are generating that connection. It could be a stretch goal to try to get data like that. Like, I mean, if they could do it in neurons, they surely should be able to do it here. But like, I'm showing you the data. This is a brand new project. I'm showing you the first steps to think about it. So we do not have that data. Okay, so maybe, because I told Adrian I would say about, so the answer is like, how do we think this thing works? We think you just need to bias contact generation towards regions of empty space. That's the key ingredient to get a network under tension. You know, I guess maybe also to answer your question, Alan, like another thing is that like, it would be really cool to make a material that did this. So even if cells don't do this, it would be fun to think about what the ingredients would be to make a material do this. So maybe that would be the rationale if pressed. Okay. Okay, so you've already asked me all of these questions, I think. So here's some of the open questions. Like, is there really something like contact inhibition of locomotion going on? Like, what is generating it? Can you really be really show that it's happening? And what's the biomolecular mechanisms that are generating that type of behavior? Is it the same as seen in sort of neural crest migration or something more canonical? What about gradients and properties along the anterior posterior axis? Because we know those are there. Like how does this thing change across that? What about this network? Yes. And the other question I have is there's a lot of other examples of sparse mesenchymal networks in lots of other tissues where there really are these large gaps between cells. So how similar is chick PSM to these other important examples? What? Lynn? Yeah. Yeah. Okay, so just to sort of now answer the question, what this paper did, and I'm going to say it really quickly, and I'm not the expert, but what they said is to achieve rigidity homeostasis. So they had this network of actin, and they have nodes, and they say, okay, we're going to remodel this network, and we're going to make new nodes. And so they have a bunch of rules in the system, some of which correspond to turnover of actin. And they claim in this paper that the key ingredient they needed to achieve rigidity homeostasis in this model was they needed to add new network edges, preferentially in direction of low existing fiber density. So that's really exactly the same thing I told you for these mesenchymal cells. And they did this very independently of us. They actually also said it helps. You need to add pre-strained edges. So you need to inject energy. So that was key. You can't have no tension. You have to inject energy via pre-tensioned edges. And they actually said it also works better if you preferentially prune low tension edges. And that actually is very similar to having P off, the rate at which you break contacts, be independent of the tension. That would do a very similar thing. So that choice seems to match up with the choice that they came up with in this system. So does that answer your question a little bit?
Yeah.
Anyway, yeah. And you, again, if you don't do these things, then you end up with a network that, like they show pictures in this paper, you end up with networks that are very clustered and weirdly and don't look anything like sort of dense actin actin remodeling networks. So I think it might be generic. Okay, I'm going to stop there. So I wanted to talk about physical learning and tunable matter and like how you can program strain in Drosophila, but I'm not going to. If you want to, I knew I wouldn't get through this, so I'm sorry. If you want to talk to me about it, I would be very excited to talk to folks here about it. Okay, you can distinguish between different ideas. Yeah, okay. Here's the summary, okay, for what I talked about. So sparse mesenchymal tissues are a fluid under tension, and well, we have evidence that they are, and they can remodel while maintaining a system spanning what looks like a system spanning tension network. And actin dynamics are similar, and maybe a word for that is rigidity homeostasis. We've developed a biophysical model that we think explains this behavior in sparse mesenchymal tissues, and maybe it might be useful for other people that want to model this hot mess of a system. We think it's a nice, simple model to do so. It does require that cells preferentially make new tension contacts. And then we can write the macroscopic properties as a simple function of key cell scale parameters.